All of you, 14 students are present in the class. Welcome. Thank you because of your presence in the holiday, Friday. So we are ready to start, and hopefully today we have planning to finish all the lectures, remaining lectures. Parasitism. So in this previous weeks, we were talking about different interactions between populations. First, we were talking about the interactions present in the members of one population, which it was competition and cooperation. And after that, we were talking about the different types of interactions present between two different populations. So neutralism and commensalism, symbiosis, mutualism, amensalism, competition, we were talking. Today it is the time to talk about parasitism. In parasitism, one way is one parasite and the other side is the host. Parasitism have some similarity and some differences with commensalism. Okay, we should start 27. So parasitism finished. And Bedelovibrio I explained, as well as myxobacteria and Legionella and these things. So now it is the time for predation, yes? Okay, predation. So it is good you are you had more progress in lecture than the other group, so we continue from this point. So predation. Predation and parasitism has some similarity and some differences. If you remember, and a few minutes back we're talking about parasitism, mostly one parasite is smaller than the host, but predator is bigger than the prey. Predator, prey, predation. The interaction between predator and prey we call as predation. So in this interaction, one predator which is bigger try to eat the smaller host or prey. In animal life, like a lion wants to eat a deer, or a lion wants to eat a mouse. So in predation, it is needed that the predator has a mouth, which normally in protozoa we call mouth as the cytostome. So some protozoa has cytostome and it is permanent, but some has temporary mouth like amoeba. amoeba has pseudopoda, so they get bacteria or any food particles and then take it inside the body like eating. But the other protozoa look like paramecium, eat bacteria by their permanent mouth. Here we have another example. As you see, Didinium is a protozoan. Didinium is a ciliate. Its body covered by cilia for movement. And this predator like to eat paramecium. So Didinium is the predator and paramecium is the prey. Didinium eat paramecium. In this figure, we can see, for example, this one is related to the paramecium, and this one is related to the Didinium. And this horizontal axis is time according to days, 10 days, 20 days, 30 days, and this vertical angle is population rate, population size. When the prey can eat and grow, they do multiplication, so their numbers are more and more and more, so the biomass is increased. When food for Didinium is more, it receive food and grow and multiply more and more. By elevation, the number of predators, the numbers of prey come down. When Didinium is the optimum number, the number of paramecium comes to the minimum. So at this time, there is not sufficient food. So Didinium decreased dramatically, and it is a good chance for paramecium to grow because the predator pressure is less at this time. Again, it is repeated, and the number of paramecium go higher. Then Didinium start to multiply and eat those paramecium. Then at optimum point of presence of predator, the numbers of paramecium come down. So it is a cyclic. And prey control the number of predator, and predator control the number of the prey. So both of them has negative control on the other one. One, the predator control the prey population, and prey population control the predator population, and it goes in such a way. So predation typically occurs when predator engulfs and digests the prey. Unlike the parasitism, which the duration of contact was long, in predation there is short duration. So in a short time, predator kill and eat the prey. So in predation, there is a short duration of contact between predator and prey. But in parasitism, there is longer time between contact of parasite and the host. And as I told you, predator is larger than the prey. Interactions of predator-prey species lead to regular, cyclic fluctuations of two populations. So these populations are ongoing in a cyclic way, and each one can control the other one. The number of predator control the prey, the number of prey control the predators. It was the life cycle of Didinium and Paramecium. Even Paramecium is the prey, but in some cases, Paramecium can be predator also. So Paramecium bursaria and the other species of Paramecium, they are predators for yeasts as well as bacteria. So Paramecium bursaria is another example, but here is not a prey, but it is a predator for Schizosaccharomyces. And here you can see Tetrahymena, another protozoa, which is the predator of bacteria such as Klebsiella and some other coliforms and gram-negative bacteria. So during the many years, many hundred years, many thousands years, predators are well equipped to follow the prey and find them and eat them. And during these times, prey understand how to escape, how to hide themselves, how they can change their appearance from vegetative form to spore form for gram-positive bacteria to save themselves in front of the pressure of predators. So there is evolution. There is evolution in predator and there is evolution in prey to continue life beside each other. Natural selection within the predator-prey system increases the efficiency of the predator in finding and engulfing its prey, but also favors those individual prey that escape predation. So nowadays, predators, not only predators, but also the prey have better adaptation for their life. Predators better equipped for finding prey, and prey are in a better way for escaping, for hiding themselves, and in gram-positive cells can changing to spore, and protozoan prey can change their appearance to cyst and save themselves. Example number 4. Interactions with clay particles provide a protection mechanism for prey bacteria. So those bacteria which are gram-negative and don't have spore, they understand they can find soil particles and going between those soils and hide themselves from the attack of predator. They hide themselves. Example, it is one example about Velexifera, which is the predator for E. coli. That is a gram-negative bacteria. It is gram-negative bacteria and can hide themselves between particles of clay. So in all of these examples, we can very easily understand that predators are so much bigger in size than the prey. For example, when predators are protozoa like ciliates, like Prometheum, when they are flagellates, like Chilomastics mesnini, for example, when they are amoeboid, regarding to different types of amoeba, they have very big size in comparison to their prey as bacteria. So bacteria are so much small in front of their predators, protozoa, which they are very in big size. And sometimes we can see, for example, in intestine, in soil, or in water, there are plenty of bacteria present. And the predator protozoan is in between a large number of bacteria. This appearance is look like a sheep in a field full of plants, which going and eating in different ways, which we call it grazing. Grazing is look like a sheep which in a field eating of different plants and going in different ways. So when one predator, protozoan, eat many prey like bacteria simultaneously and going in different ways, we call it grazing. Predator grazing. But as I told you, even though some bacteria, mostly gram-negative bacteria, hide themselves to avoid to be eaten by predator. Some bacteria, especially gram-positive bacteria, mostly they are equipped to change their appearance from vegetative form to a spore form. So sometimes, even though for predator, as food is sufficient, pH is okay, temperature is okay, so we can see all the things are all right, and we feel they should be vegetative form. But because they are under the pressure of predators, or even though under pressure of the parasite, they change their appearance from vegetative form to a spore. So spore formation is a mechanism for escaping from pressure of the predator. Some microbial structures are resistant to predators. For example, soil amoeba engulf vegetative cells of bacillus. But if bacillus change its shape from vegetative form to spore, so those amoeba cannot engulf the endospores. So vegetative forms of bacillus is food for amoeba, but their endospores not known as food, and they can save their life by disappearance and waiting till the time that pressure is finished. The predator pressure to be finished. Okay, at this part, so now we can say that approximately we explained completely the different types of interactions present between two populations. First, we explained the different interactions between members of one population. It was only competition and cooperation. Then we explained the interactions present between two different populations. We explained it could be neutralism, commensalism, symbiosis, mutualism, and competition. Between two different populations, we start for a new subject related to microbial communities and ecosystem. Microbial communities and ecosystem. And there is one subtitle. Our subtitle is population selection within the communities. What does it mean? It means we want to say, are all population have the same power, or some populations in an ecosystem has more power. If some are more powerful, if some populations are more powerful than the other population, why they have more power? The power in population obey the rule of RK strategy. RK strategy. R is the abbreviation for high reproduction. So any population which can reproduce faster, so the most probable it is the winner group. Suppose there are two teams, they are playing with each other. They start a play one group every 1 minute, one person is added. And the other group, each hour, one member is added. So after 60 minutes, one group is composed of 2 persons, and the other group is something near to 60 persons. So in any struggling, in any fighting, in any interaction, when one group, the number are so much more than the other group, so the winner is the group with more members. So population is the power. Population is the power. And the other item is K. K, or physiological adaptation to environmental resource. There are different resources, different types of foods. Suppose cellulose, pectin, chitin, hemicellulose, starch, and so on. There are many different foods present in the environment. So those microbes which like all the present nutrient in one environment, they are winner. For example, suppose 2 children are present at one home, and whatever mother prepare and cooking, one child says, I like it, I like that, all the foods are good, and the other says, I don't like this, I don't like that. Who is happy to live on that habitat? So the members which adapt to whatever food is present is winner, and the population which is not adapted to the nutrients is loser, the game. Now these 2 factors come together. So any population which can multiply faster and adapt to use whatever is present in the environment is the winner and is more powerful. After a few minutes, then I explain if one or some populations have the best RK strategy, they are in the best way obey the rule of RK strategy, and they have best R and best K. Why they exclude? Why don't they exclude the others and alone to live there? So wait, then after a few minutes I explained it. We have another item with the name of succession. Succession. Succession means changing the member of community, changing the populations in one community. For example, suppose some microbes are present in one habitat. After some time, some are removed and the new ones are coming. So we are talking about succession within microbial communities. We have communities and we want to see how some new microbes are coming and how some old microbes are omitted from the environment. In order to understand in a better way about this subject, we should find one habitat empty of any microbes, and then we follow which microbes are coming there at first, then which second populations come there and previous one omitted, and what are the final groups, which members of microbes, which populations can be permanently present on that habitat. One good habitat for this experimental studies is human and animal intestine. Human and animal intestine. So as you know, when a fetus is in the uterus of a mother, it is completely sterile. After birth, the newborn, by first breathing some bacteria, come to the upper respiratory tract. By touching the newborn, first microbes can be present on the skin. And afterwards, by first drinking milk by mothers, and first microbes can be present in the intestine. So when we have a habitat without any microbe, we call this environment as virgin environment. Virgin environment. Virgin environment means an environment empty of any life, empty of any microbe. So when newborn is drinking milk, the first microbes are present there. The first microbes present in any environment, in any habitat, we call them as pioneer organisms. Pioneer organisms. So pioneer organisms are those microbes which they are present at first microbes in one habitat, and they start primary succession. In a place without any microbe, the first microbe are coming and settle down. We call them, they produce primary succession. But after some time, along with milk, another food, for example, boiled water, then slowly soap, then a little bread are added to food of the child. So with new foods, new microbes, new populations are coming to the intestine. Even though sometimes some of the pioneers are omitted and the new ones are settled down there. So now it is secondary succession. Pioneer microorganisms alter condition in a habitat in a ways that discourage further succession. So, population better adapted to the now colonized usually replace the pioneers. As these habitats undergoes additional changes, secondary invaders are also replaced. So the animal or the child slowly grow. And if we look to the week, month, and year food, they come to a constant foods. And every month, every year, they have some repeated foods. So those microbes are present in the intestine. They have constant food, and they come to a final community. Final community or climax. So after primary and secondary or maybe third, fourth succession, by the way, we reach to the climax community. In this case, succession ends with a relatively stable assemblage of population, which we call it climax community. So climax community is achieved. But climax community almost is or mostly is theoretically, because in any case sometimes there are some changes, some drug using or something else changing the community and population present in the intestine. But by the way, approximately we have constant microflora in our body. But it is not so. All human or all animals have similar, completely similar community. Because, for example, some people are vegetarian, their foods completely are dependent on eating plants. Some people are pure non-vegetarian. Some people have the mixture of food of vegetables as well as meat, with fish, with the other sources. So, which sources of food one person is using during life force which populations and which community should be dominant and should form the climax community, should form the climax community. Here we are talking about succession. Succession, as I told you, means changing the populations, in one community. Sometimes changing the types and numbers of populations present in one place is related to the action of some live microbes. And sometimes it is not completely dependent on the presence of those microbes. If succession is related to the action of any microbe, we call it autogenic succession. If succession, again telling you, if succession is related to the action of one population or one microbe, we call it autogenic succession. It is autogenic succession. For example, creation of anaerobic condition by facultative anaerobes allow development of strictly anaerobic population. Suppose when there is a newborn child, a little oxygen is present in the intestine of newborn. So obligate anaerobes cannot to be as the pioneer and cannot live there. But first facultative anaerobes, which first they go through the oxygen and using oxygen remove all the oxygens are present in the intestine. And accordingly, now the situation of intestine is completely suitable for growing of anaerobic bacteria. So if anaerobic bacteria couldn't live there and now can live there, it is because the action of facultative anaerobes. So we call it autogenic succession. But if succession is not related to the action of any microbe and it is mostly related to the environmental factors, we call it allogenic succession. Allogenic succession. For example, it occurs when a habitat is altered by environmental factors such as seasonal change. Suppose now it is summer and in the river mesophilic bacteria are present. So by changing the season, when winter is starting, so the water is more cold and the temperature comes down something around 2-3 degrees centigrade. So mesophilic bacteria which find the situation is not good, mostly they go to the sediment to save themselves. And from now on, the dominant microbes in the river are cyclophils. So it is a succession. In the winter, mesophils are going and cyclophils are coming. This succession was not related to the action of one microbe. It was related to the environmental changes. So if environmental changes is responsible for succession, it is allogenic succession. If environmental changes, environmental factors are not responsible for succession, and succession is related to the activity of one microbe, it is autogenic succession. Autogenic succession. There is one example over here, succession of microbial communities related to sterile gastrointestinal tract. Our aim is not gastrointestinal tract, but gastrointestinal tract of newborns because is virgin, as it is a good habitat for experimental study, we focus on it. Okay, so we are studying gastrointestinal tract of newborns because in newborn it is a virgin habitat and experimentally we can follow what happens in this environment with succession happens in this environment. Population level and types of microbes in climax communities are regulated by multifactorial processes. And some of these regulatory forces in this process are extracted by these items. So many factors or multi-factors are present, which microbes should be in one habitat, and why and when some microbes should remove and new microbes should be replaced. First item is animal host. First item is the animal host. What is that? So when animal hosts are different, for example, suppose a lion intestine, a cow intestine, a fish intestine, when those animals live in different places and they have different intestine. So intestine now is the habitat. When habitats are different, so we should wait to see different microbes, different populations, and different communities. So animal hosts, when they are different, they have different alimentary canal, so different microbes should be present there. Because their environment, the habitats are different. The intestine of a cow is so much different with intestine of a fish, and it is so much different with intestine of human. As much as the habitats are different, the populations and those interactions are different. Another way, not only the habitats as intestine of different animals are different, habitats are different, but also their diets are different also. Not only in ruminants we can see rumen, and in some animals like horse we can see a large cecum, and in human another appearance, but also the food of fish which come to the intestine is completely different with the food of horse, and it is completely different with food of human. So what is the food for one animal or one human is the food source for those microbes present in their intestine. So diet is directly correlated with the types and the numbers of microbes present there. According to the types of intestine and types of diets, some microbes are present in the intestine. So when some microbes are present in intestine, they go to find which members on the community are native and which microbes are not native. So all microbes, all different microbes in the community, they fight with other microbes according to their own food and space. So microbes are responsible. Which other types of microbes can be present? For example, in our intestine, some microbes which are present in the cow intestine is not present, because the types of microbes present in microbial community in our intestine completely are different and actively respond to other microbes coming to the intestine. And environment. So environment is a place which animals find food, and which types of food can be found by any animal is the food for microbes and microbial communities present in the intestine. So environment is very effectively affect the types and numbers of microbes present in the intestine. There are many niches filled by various microbial populations. What is niche? Suppose when we talk about a habitat like ocean, some upper parts is containing full of oxygen and full of light. So many photosynthetic microbes and algae are present there. When we go to the deep sea, there is no light and there is no oxygen, and it is completely darkness. So those microbes which could live in the upper side never can live in the deep sea. So we can see this is a habitat, common habitat, but it has several different places in one habitat, which we call them niche. Like one apartment or one home have several rooms. Some are in one room, the other in another room. One place is the kitchen, one place is the hall. Even though it is one home is a habitat, but it has different places. Different places on that habitat are present, which we call them niche. One room is related to the child, the other room for parents, the other room for sitting room, the other for kitchen. Okay, so one place like a river, soil, ocean, or whatever have several parts, several locations, which functionally microbes can live there. Microbes can affect the environment, and environment can affect on those microbes. So we call it niche. We call it niche. There are many niches filled by various microbial populations. Succession of bacterial population in gastrointestinal tract of human and non-ruminant beings. We want to see how much is the similarity between the microbes in non-ruminant. Ruminant look like cow. but now we focus on non-ruminant non-ruminant animals, and those microbes starting to have succession in their intestine. First, the pioneers are Bifidobacterium and Lactobacillus species. After some time, facultative anaerobes are the dominant, especially coliforms such as E. coli and also Streptococcus faecalis. Then by presence of these facultative anaerobes, whatever oxygen is there is removed, is finished. So from now on, it is the time for presence of strictly anaerobic bacteria, such as Bacteroides, especially after solid food ingestion. Obligate anaerobes become dominant. Climax microbial communities in gastrointestinal tract are generally reached by the time of the weaning. Or another example to see the succession is related to the mice. It has some similarity and differences than before. The pioneers are Flavobacterium. Then after 8 days, Flavobacterium disappear, and Enterococci and coliforms increasing dramatically. Then lactic acid bacteria increase for 10 days, after which remain constant. After 18 days, dramatic increase in population of Bacteroides, which become dominant population in climax community can be observed. Okay, so now we want to go to the new topic of this part, and we have sufficient time to finish today. So if you accept, we have a break, something around 20 minutes. And 4:30, again, we will continue our lecture, but we should leave the lecture and we return back to the new class. So the quality and quantity according us and according to the university to be acceptable. So if you agree, we are going for 20 minutes break. Then